Picture a single droplet of pond water glistening on a microscope slide. Inside that speck, a lone microbe that's quietly thumbing its nose at the rulebook of life. Here's the headline: the usual DNA stop signs, the genetic periods that tell a cell, all right, end of sentence, don't mean stop at all for this organism. Biologists have long called that code nearly universal. Turns out, it might be more of a helpful suggestion. Now, how did anyone spot this rebel in the first place? The story starts with some sequencing data that looked wrong. Think of a photo download that freezes halfway. Pixels everywhere, nothing lines up. Same vibe. But when the team reran their tests and then used completely different tools, the odd pattern refused to disappear. Signpost 1. What exactly looked strange? 3 tiny DNA words, triplets we normally read as a hard stop, were showing up smack in the middle of long, well-built protein plans. If those really were stops, every protein would be shredded nonsense. Yet the microbe was alive, swimming happily under the lens. Something didn't add up. So the researchers slowed down. Instead of labeling the data an error, they treated every oddity like a clue. First, they zoomed in, single cell level. Then they zoomed out, dozens of related cells. Was this a rare glitch or part of a wider trend? The deeper they dug, the clearer it became. This wasn't a few typos. It was a systematic rewrite of the punctuation marks of life. Time for a reality check. To prove the cell was truly reading past those stops, the team hunted for the actual proteins being built. Mass spectrometry, basically a molecular fingerprint scanner, showed one building block popping up over and over: tryptophan. Normally, tryptophan uses a completely different genetic word. Yet here it was, slotted in where a stop should be. That's like seeing a green light where every traffic manual swears there should be a red. And that raises the big question: how does a cell survive a rewrite this deep without melting down? The answer lives in those tiny adapter molecules, the tRNAs, and their partner proteins. Imagine swapping both your keyboard layout and your autocorrect rules at the same time. Total chaos if you change only one. Weirdly functional if the whole system shifts together. That's exactly what appears to have happened. Signpost 2. Is this microbe a lone outlaw? Not even close. Surveys now hint at dozens of independent code rewrites sprinkled across the tree of life. Some lineages convert one stop into a signal for a rare amino acid. Others make the stop behave like a dual personality, sometimes a period, sometimes a comma. The so-called standard code may just be the most popular dialect, not the only one. Need a mental image? Think about watching a foreign film with subtitles that almost, but not quite, match the dialogue. Most lines sync perfectly, but every so often the break hits early and the next sentence rushes in. That's what's happening inside these cells. The timing depends on the broader sentence, not a single 3-letter cue. Why does it matter beyond fun trivia? 2 huge reasons. First, biosecurity. If nature can run alternative genetic dialects, synthetic biologists could engineer private cellular languages, built-in safe rooms, where invading viruses, still speaking standard code, can't hijack the script. Second, evolution. Many tools that draw family trees assume stop signals are fixed. If those assumptions wobble, some of our tidy timelines for when species split might need a serious redraw. Signpost 3. This discovery even nudges the search for life beyond Earth. Datasets we once binned as corrupted might hide organisms playing by unfamiliar rules. Future software may scan raw sequences the way Shazam picks out a remix inside static, flagging tracks that use familiar notes but dance to alien rhythms. Let that sink in for a moment. One microscopic rebel, sitting in pond water, just expanded the boundaries of what we consider possible genetics. Before we wrap, let's ground this in you and me. I've got 3 quick reflection prompts. No need to write them down, just chew on them during your next coffee break. 1. Where in your own work or your daily routine are you assuming the rules are fixed? What's your personal version of the DNA code you've never questioned, and how could you safely test an alternative? 2. Scientists can add extra letters to DNA and the cell still hums along. So, what extra letters, maybe a new skill, a fresh perspective, or an unexpected collaborator, could you weave into your current project to unlock something you've been told is impossible? 3. All innovation carries risk. How do you draw the line between bold creativity and going too far in your field? What guardrails can you set up this week to stay on the right side of that line? So, here's what we covered today. First, a humble pond dweller flipped a genetic stop sign, proving the code of life isn't as universal as we thought. Second, the cell survived because its whole translation toolkit, those tRNAs and helper proteins, shifted in sync, like changing both keyboard and autocorrect together. Third, this isn't an isolated quirk. Dozens of species play with their genetic punctuation, which reshapes everything from evolutionary timelines to biosecurity strategies. And finally, that flexibility opens doors. New ways to block viruses, fresh clues in the hunt for extraterrestrial life, and a reminder that impossible often just means untried. Take that mindset into your week. Question the stops you assume are permanent. Rewrite a small rule of your own, test it, and see where the new reading frame takes you. Until next time, keep exploring the edge of what's possible. And remember, even the tiniest cell can teach us to read the world in a brand new way.